Yeah. So good morning, students. I hope you can hear me, see me, and you can see my PowerPoint. So let me start the class. So this is a immunohistochemistry or advanced histological techniques. So and the subject code is 21 MSH 716. So currently we are in the unit 1. Unit 2 has been finished, immunohistochemistry, and in the unit 1, the major topic is enzyme histochemistry. So the idea of this enzyme histochemistry is to visualize the enzymal activity within the tissues. So here we are going to study the enzymal activity within the tissues because this enzymes plays a vital role in any organ normal function. So not only seeing the proteins within the organ, okay, in order to assess its status, working status, we also need to measure the enzymal activity, and that will give us a precise state of functional state of the organ. Okay, so that is why we are going with this enzymal studies, and that is the idea of this enzyme histochemistry. So here we are trying to localize the enzymes present in the tissue. Okay, and we have many types of enzymes, and those enzymes has been specific to many organs like LDH1. So these are lactate dehydrogenase isoenzymes. Like LDH1 is present in heart, 2 is present in reticular endothelial system, that means white blood cells, 3 is in lungs, 4 is in kidneys, 5 is in liver and the striatal muscles. Okay, so yes, enzymes have their own significance, and measuring them will give us some idea about the status of any working status of any organ. So this is the introduction to enzyme histochemistry. Then, according to the syllabus, we need to study all these 5 enzymes. We need to study phosphatases, dehydrogenases, oxidases, esterases, and tyrosinases. So before going to study individual enzymes, we need to have some basics. So basics about enzymes. This basics we already covered in the last lecture, but anyway, I will do a quick revision. So basically, the idea in enzyme histochemistry is in order to assess any enzymal status, we will see the okay. I want whiteboard here. Uh, just a second. Let me take whiteboard. 1 second. I need to draw. Then you will understand how we are doing. So let me take. Yeah, now you can able to see whiteboard, I hope. So let me show you what is the enzyme histochemistry. So let me take one highlighter. See students, so we have a tissue, okay, and in this tissue, say for example, we have a enzyme. We need to see the functional status of the enzyme. So I'm showing enzyme like this. Okay, so this is the enzyme. Okay, let us assume this is LDH enzyme. Okay, lactate dehydrogenase enzyme. So this is LDH enzyme. Now, how we will detect this enzyme means we are not going to detect enzyme directly. Rather than now we will add substrate to this enzyme. Okay, so I'm going to add one substrate. Okay, specific to this enzyme. Okay, so I'm showing in 2 colors, so you will understand. So this substrate is a 2 molecule entity. So this is a 2 molecule entity like this. It has a green and blue. Okay. So now I added this substrate to the enzyme. Okay. I added this substrate to the enzyme. Okay. I added substrate to enzyme. Now this enzyme will break down the substrate into product. Okay. It will break down substrate into product. So this is the 2 products. We got 2 products from the enzyme. Okay. So we got 2 products. So now we will take any one product, and now we will try to colorize this product. We will try to react this product with any secondary substance. So to this product, I will add a secondary substance. Okay, I will add some coloring agent. I will add some other chemical like DAB. Okay, just example. So I added DAB. Okay, this product also don't have color. Keep in mind. So this is substrate, this is product, and product don't have any color. Now I'm going to visualize the product by adding some secondary substance. Okay, here in case here I added this diaminobenzoate. Upon adding this substrate, okay, upon adding coloring thing, now I will get a color. So this will give yellowish color like this. So now development of yellowish color indicates presence of our target enzyme within the tissue. Now, here this enzyme histochemistry will give us 2 data. One is qualitative data, either presence or absence of a particular enzyme in the tissue. And second one, the quantity of the enzyme present in the tissue. How many enzyme molecules present in the tissue? And that number of molecules present can be detected by the intensity of the color developed by the upon adding the substrate. The higher the color, the more the enzymes present in the tissue. So the intensity of the color developed will give us the quantity of the enzyme, whereas the presence development of color itself indicates the presence of enzyme within the tissue. Okay, so this is how we are going to measure enzymal activity indirectly by adding substrate. The higher the color production, the better, the higher the molecules of enzyme present in the tissue. Okay, so this is the basic idea of enzyme histochemistry. We have a target enzyme reacted with substrate, substrate broke down into product. Now this product will be visualized by various techniques. Okay, sometimes we will use salts. Okay, sometimes we will use a diazo salt to visualize the product. Sometimes we will use coloring agents. Okay, naphthenols. So there are four principles and four specific reactions to visualize the products. Okay, we have four principles and four reactions to visualize the product. So now I will discuss about what are the four principles and what are the four specific reactions we have in the enzyme histochemistry. All enzyme histochemistry techniques are based on these four reactions only. Okay, these four principles and four reactions. Okay, so now let us see what are the four reactions and these principles in enzyme histochemistry. So meanwhile, let me do a quick revision of the basics of enzymes. So enzymes are something that will speed up the chemical reaction. They will react with a substrate and they're specific in their nature. One enzyme will only react with one particular substrate. It's like a lock and key mechanism, just like your antigens antibody. So that is the thing about enzymes. Now, the properties of enzymes include they will speed up the reaction. They unchanged during a chemical reaction. All enzymes are proteins. They are reversible. Upon participating one enzyme within a reaction, it can be reutilized for the secondary reaction. Okay, and also there are certain factors that will affect the enzymal activity. So factors that contribute to enzymal activity include concentration of enzymes, substrates, products, temperature, pH, activators, inhibitors, time, presence or absence of radiations, oxidizing agents, and anti-enzymes presence in the tissue will they are contributing to the uh, the rate of reaction we are developing. So these are the few factors that affect in the enzymal activity. So we need to consider these factors also when we are performing this enzyme histochemistry analysis. Okay. So all these are you already. Then I discussed about various classifications of enzymes. We have reductases, transferases, hydrolysases, lyases, isomerases, ligases, and I have given their typical reaction and their example also we have studied. Okay. So this is the basic classification of enzymes. Since we already discussed this, I'm not going depth to uh, restudy these things. Okay. Then coming to enzyme histochemistry. So here, as I stated, same story. Enzyme histochemistry is a technique where an enzymes within the tissue will be studied by some chemical approaches. That is called enzyme histochemistry. So significance of enzyme histochemistry. Very simple. All organs require enzymes. So if there is any problem in the enzymal activity, definitely that organ will face some dysfunctions. Say, for example, lactate dehydrogenase. Lactate dehydrogenase is very important for normal activity of heart. If the enzymes within the heart has any abnormalities, then definitely there will be a possible ischemic heart attack or cardiac abnormalities can be developed due to failure of or malfunction of the corresponding enzyme. So yes, enzyme histochemistry has a huge significance in clinical diagnosis of any disorders. Okay, yeah. So then histochemical reactions, uh, as I stated, substrate enzyme is present in the tissue. Then upon adding substrate, now this will make a primary reactive product. I said right, we develop the product, but this product don't have any color. If product don't have any color, now we need to add certain captures, so certain capture molecules to visualize the product. Okay, so we will add certain capture molecules, and this capture molecules will give us a final reaction or final coloring reaction will be developed. So this is the basic idea of immunoenzyme chemistry. Okay, so it has ample of applications in clinical diagnosis. Then yeah, see, I given one example here, enzyme. Yes, ma'am. So we have enzymes. enzyme in a tissue, we added a substrate, it given a primary reactor to product, a product. Now, in order to visualize this product, I added diazonium salt. In the previous slide, I said, right, I added a salt to the product. Now, the salt will react with the product to give the final coloring outcome, okay, final colored product. So this is the basic idea of enzyme histochemistry. Yeah. So there are certain factors in enzyme histochemistry. Keep in mind, now I'm going to discuss about the factors contributing in enzyme histochemistry. I'm not discussing about factors of normal enzymes. Okay. I'm not discussing this in the view of biochemistry. Rather than I'm discussing this from the point of enzyme histochemistry. So some factors that contributing to lack of coloring or lack of report generation, including material processing. If we haven't unfixed after fixation, if we are not removing crosslinks properly, there is a possibility of not getting any results. So that is one factor. Then temperature. Every enzymes require a optimum temperature for their normal activity. If we increase the temperature of this tissue, definitely these enzymes not going to react. So temperature we need to keep in mind, and pH levels. Every enzyme has their own optimum pH ranges for their functional activity. So any fluctuations within the optimum pH can result in diminishing of our enzymal activity. So we need to keep an eye on pH also. Then a concentration of enzymes and concentration of substrates and concentration of products are we need to keep control of them also. Products react substrates and enzymes. Okay, even they can influence the results. And inhibitors. Sometimes during the fixing procedure, we need to inhibit the normal enzymal activity. Why we need to inhibit enzymal activity? Because if they are keep on reacting in the tissue, they might alter the results. Okay, so sometimes in some procedures we will purposefully block the enzymal activity in order to preserve them, in order to get more accurate data. So that might happen in some tissues in some studies. Okay, so definitely we will add certain inhibitors. So inhibitors role is to inhibit the enzymal activity. Okay, we can restart the enzyme. Okay, inhibitors are reversible, so we can remove inhibitor to get the normal enzymal reactions. Okay, and yeah, so all enzymes require certain cofactors for their which will act as activators. Okay, sometimes even after adding substrate, enzymes cannot start working because they still dependent on certain cofactors like magnesium, calcium. Only in the presence of this cofactor, an enzyme will bind with the substrate. So sometimes we will add these activators also. Okay, the activators can be stated as a cofactors. Okay, yeah. And controls are very important. It is because of these controls we can able to validate our test results. So controls are very important. Irrespective of any test, controls have their own significance. Okay, yeah. So fixation can be done either by by using enzyme hydrolytic enzymes, or by heating procedure we can do the fixation, just like our immunohistochemistry. Okay, so I'm just skipping this fixation thing. Same fixation. Okay, the best method of fixing will be always cryofixation only. Okay, cryofixation. This procedure will minimize the enzymal damage. Okay, so cryopreservation and cryofixation has better results. Okay, result outcomes. Yeah. So we can make smears, we can take biopsy, we can make smears, and we can go for this enzymal studies. Okay. So coming to these reactions and principles and reactions. So this is very important. This is our today's lecture. So pay utmost attention from now. Okay. What are the principles and reactions involving in enzyme histochemistry? Are you following me, students? Is everyone listening me? Yes, students.
Yes, sir.
Yes, perfect. Okay, thank you. Yeah. Now see, we have four principles. One is simple ionic interactions. Okay. One method of reacting enzyme with substrate is by ionic interactions, and other principle is by reacting with aldehydes with a Schiff reagent. So by reacting the aldehydes within these enzymes with the help of shift reagent, we can able to visualize the enzymes, or by using diazonium salts. Okay, so we will get the product, and this product will be subjected to diazonium salts, and those salts will give the residue, the coloring residues. So by using certain diazonium salts, we can able to visualize the enzymes, and by conversion acting on substrate to form a coloring repeat. Sometimes directly this substrate will be developed into a product, coloring product. It's a one-step procedure. We will have substrate, and substrate will undergo it will be break down to product, and product will have color without a subsequent addition of anything. The substrate can itself able to develop color. So that is a substrate to form color precipitate. Okay, so these are the four reactions. We have ionic interactions, aldehydes reacting with aldehydes with shift reagent, or with subjecting by diazonium salts, and this substrate itself will develop the colors. Okay, so now these are the four principles on how an enzyme will react with substrate. So these principles are discussing about the enzyme substrate interactions. Now the reactions, the chemical reactions are discussing about how to visualize the product. So these reactions we can say as a capturing methods. Okay, so enzyme reacted with substrate and it given a product. Now how we are going to check the product, how we are going to visualize the product. So the product visibility can be done in these four reaction methods. Okay, we have simultaneous capture method, post incubation coupling method reaction, okay, self color substrate reactions, and intramolecular rearrangements. So now let us discuss in detail about these four reactions. Okay, so the first one, as I stated, simultaneous capture. Okay, so simultaneous capture, as the name suggests, it will simultaneously capture the enzyme. It will develop a substrate, and substrate will give the color. Okay, so yeah, so the idea of simultaneous capture is we have enzyme reacted with substrate. Okay, they given PRP, primary product reacted with a salt, and salt given as a this one. What is this? Salt given as a immediate color. So this is called a simultaneous capture. So immediately after developing primary product immediately reacted with this diazonium salt, and it given the product. So this is the idea of simultaneous capture. So it has its own disadvantages. Okay, we have this diffusion of a primary product. Okay, the chance of getting accurate results is less by simultaneous capture technique. Okay, I will show with visualization like this. Yeah, a diagrammatic representation of simultaneous capture. So we have a enzyme substrate, we added salt, so they made a primary product. Okay, substrate enzyme product, and simultaneously they reacted with diazonium salt, and they given a coloring component. So the advantage of simultaneous capture is a great repository for alternative substrates and highly sensitive. So this method is highly sensitive, but since it is highly sensitive, there is a chance of getting false positive results. Even though there is no substrate within the tissue, but still it will give us a false positive results. So that is one major disadvantage of this simultaneous capture. Okay, then what is the next technique? So the next technique is post-incubation coupling. So what is this post-incubation coupling? So let us see. So in post-incubation coupling, we added enzyme, and we already have enzyme. We added substrate. So they given primary reaction product. Okay, enzyme substrate product. Now they become insoluble and remain at site of production. Okay, so upon reacting substrate with enzyme, they will make an insoluble component. Okay, it made a precipitate, an insoluble precipitate. Now this insoluble precipitate will be filtered, and it will be re-incubated with secondary coloring agent. So that is why this is called post-incubation coupling. So because the outcome of this particular reaction is insoluble substrate, now we will filter this insoluble substrate, and we will go for incubation. Okay, initial incubation is necessary. We need to incubate this insoluble substrate in this solution, so that will give the color. So that is the post incubation coupling, and I will show the diagrammatic representation. Yeah, so we got an enzyme, we got a substrate, They made an insoluble component. Now this insoluble component will be subjected to the secondary. Okay, we filtered them and we subjected to diazonium salt, and it given the final product. So instead of a simultaneous capture, here they made an insoluble component. Now this insoluble component need to be filtered and they need to be subjected to coloring procedure. So this is the post-incubation coupling. So after incubating, we are coupling with some other thing to develop the color. Okay, so this is second type of reaction. So first one is simultaneous capture and second one is post-incubation coupling. Okay, so it has advantages. So case where long time incubation stage is necessary, optimum pH for enzyme and diazonium salt can be separated easily. So for in some tissues where we need longer incubation times, for that post incubation time is the better approach. And a disadvantage, same like sensitive only. I guess this post incubation also we are using diazonium salt. Maybe this is highly sensitive and it might give us a false positive results. So that is the disadvantage of this one. Okay, and another disadvantage, students, sometimes this primary product will be diffuse. It is not stable component. This primary product, even though they binded enzyme binded with substrate, it will easily diffuse. If it is immediately diffused, if it is not going to react with our secondary product, then it is useless. So that is another disadvantage of major this enzymatic studies. Okay, this is called a diffusing of primary product. So this is all about post-incubation coupling. Then let us see the third one. So what is our third one? Self-colored substrate. So what is this self-colored substrate reaction? So we have an enzyme, we added a substrate, and substrate that immediately given us color. Okay, so just by adding substrate itself, we got coloring product. So there is no second step. Secondary step is not necessary for self-coloring substrate. So that is the idea of self-coloring substrate. Okay, substrate itself will react with enzyme to give the color. So let us see the chemical reaction. So we got an enzyme, we added substrate. Substrate immediately given as a coloring product. So this is undergone by hydrolysis. Because of this hydrolysis property, the substrate itself will give us a color. Advantages: diazonium. Okay, salt coupling is not required. Disadvantage: only few substrates will act like this. We have a limited availability of substrates which have this property of self-coloring substrates. Okay, and we need not to go for secondary step, adding of salt in this step. Okay, so this is the major advantage of self-coloring substrate. Okay, so this is third reaction. And the last and final reaction is intramolecular rearrangement. What is this intramolecular rearrangement? So we got a soluble okay. We have an enzyme. This enzyme will be reacted with a soluble substrate. Okay, so this soluble substrate will rearrange the spatial structure. How can I say? You know what are isomers? You know, they will rearrange the structure of the molecule by rearranging the structure of molecule. Now we got a different product, and that okay, it's a different product of the same component. Rather than it is in a D glucose, now it is in L glucose. Okay, isomerization. So it rearranged the molecules. The spatial structure has been just rearranged by this substrate. That's it. It haven't done any chemical reaction. Rather than it just twisted the molecule. Okay, so this molecular rearrangement can happen, and that can be reacted with the secondary product to get the coloring component. So this is the fourth reaction, fourth type of reaction to visualize the enzymes. Okay, let me show diagrammatically. So we got an enzyme, we added a soluble substrate. Now it rearranged the substrate molecules, then it given a color upon secondary addition. Okay, so here also there is no salt requirement, and only limited substrate have this property. So these are the four reactions of enzyme histochemistry. So a quick summary of these four reactions and principles. So the principles are the enzymes will substrates will bind with enzymes either by simple ionic interactions or due to aldehydes with Schiff reagent or coupling with the salts and conversion of substrate itself will develop color. So now in the next four slides we have observed that diazonium salts played a crucial role in enzyme substrate reactions, and also self-coloring substrates also played a crucial role. Apart from these two, the Schiff method is most commonly used in labs, okay, because Schiff reagent is also used in some other tests. So that is why, since it is easily available, we have many methods based on the principle of Schiff reagent also. Okay, so these are the four principles and these are the four reactions: simultaneous capture, post incubation, self colored substrate, and intramolecular rearrangement. Okay, so simultaneous capture, this one. So here the diazonium salt played a crucial role. Then second one is post incubation. So we got a primary product, and that is insoluble product, and this insoluble product has been incubated to develop the color. So that is post incubation coupling. Then we discussed about self colored substrate. The substrate itself will develop color. And last and final, we discussed about the this one. What is it? Intramolecular rearrangement. It will rearrange the intramolecules of the substrate so that upon secondary subjection, it will give us the color. So this is all about the enzyme histochemistry. Do we add insoluble substrate in post incubate? Do Jasmin, they're asking, do we add insoluble substrate in post incubation coupling? 1 second. We are not going to add that. That is already developed. Okay, let me show you here. See, enzyme substrate, they themselves made insoluble substrate, insoluble component. Now, to this insoluble component, the salt will react. Okay, we can say one thing. This insoluble substrate is not so stable. It is weak insoluble substrate only. Okay, so this can be easily reacted with the diazonium salt. Whereas here, the substrate is very strong. Uh, yeah, that's it. 1 second, I was confused here. 1 second. Now, when we see the difference between simultaneous capture and post incubation, in simultaneous capture, the bond is not so it is not so strong. Okay, so that is why it quickly reacted with salt. Whereas in post incubation, the bond is very strong, so that we need to keep it for a long time. We need to incubate this insoluble substrate or product for a longer period of time. When I say longer, just half an hour. So we need to incubate this for half an hour in the diazonium salt to get the color. So if there is a strong bonding, then we will go with this post incubation coupling approaches. That's it. Okay. Post incubation coupling occurred within the tissue only, because there is a very strong interaction between the enzyme and the product. Okay, so that is why here we will subject it to long time with the salt to develop the color. Yeah, so I cleared your doubt, right? So yes, in simultaneous capture, the reaction is very fast, whereas in the post incubation, the bonding is very strong. So in order to develop color, in order to break down this product and to develop color, it will take more time. So we need to keep it for incubation to develop the color. That's the only difference between simultaneous and post incubation coupling. Then self colored substrate we already understood, and intramolecular rearrangement also we already understood. Okay, so yeah, so it depends on what type of substrate we are using. Correct. Yeah. Yes, Jasper. Yeah. And also, usually students, we have this tough substrates only. The most commonly available substrates are not so friendly. Okay, we have seen, right, like our self-colored substrate and intermolecular rearrangement substrates are rare. Actually, by using them, we can do test very fastly. But because of their rarity and their limited availability of only few range. Okay, for only few enzymes, these substrates are available. So that's why we are going with this salt approaches only. So this salt can able to react with any enzyme, any substrates. So we are trying with traditional approaches. Actually, nowadays enzyme histochemistry is not using. Rather than we are going with advanced techniques like flow cytometric analysis in FACS. Okay, we have FACS and other advanced techniques are available. But the only advantage of enzyme histochemistry is low cost. Okay, for a FACS test, it will take around 5,000. For enzyme histochemistry test, it will take around 500. So there is a drastic variation in the lab cost. Okay, this test cost. So in any small primary healthcare or simple government hospitals, we will try with enzyme histochemistry only rather than this FACS analysis. FACS machine is very costly. Okay, Yeah, that is one reason. Yeah, yes, students. So this is enough for today's class. In tomorrow's class, we can start discussing individual enzymes. Okay, like how to detect various types of enzymes like alkaline phosphatases. Those things we will see in the tomorrow classes. Okay, uh, I think within 2 lectures we will finish this enzyme histochemistry. So I will do a quick revision, then we can wind up. Okay. So in today's lecture, we discussed about the factors influencing the enzyme reactions, and we discussed in detail about the principles and specific reactions in the enzyme histochemistry. So the 4 principles including ionic interactions, aldehydes with Schiff reagent, diazenyl salts, and by self-coloring substrate. These are the 4 principles, and the 4 reactions include simultaneous capture, post-incubation coupling, self-coloring substrates, and intramolecular rearrangements. Okay, so this is enough. This is it for today's class, students. I hope I already cleared your doubts. If you have any more doubts, you can ask me. If not, we can wind up. Can I end this class, students? Any doubts in today's session? I hope no doubts, right? So I made a nice animation also. Individual enzymes, we will go with animation. You can easily understand, okay? So I will ensure. And also feel free to ask any questions, any doubts, okay? So I won't tell anything. I won't scold you, okay? I like if you're asking more questions. And also I really appreciate the one who asked questions like Jaspinder and our Jaspinder only, Just Karan and Karan, Just Karan. Okay, he asked about the Jaspinder is active listener. Great. Okay, thank you very much, students. You can leave. Meet you tomorrow. Okay, see you tomorrow in enzyme histochemistry. Okay, see you, students.